All right. Well, hello, everybody. It's great to be back in Kansas again, or wherever you are. I'm Cheryl Stucky. It's a real pleasure to be asked to give this talk on STEM and on things that I've done in my career. I'm so delighted that Dwight asked me to speak. So I'm just going to tell you a little bit about my career path and some of the things that I do right now. We're not in Kansas anymore, as you see from my yellow brick road in Dorothy, but it was a fantastic place to grow up. And I will say that the number of times I have been asked by people who are not from Kansas whether I know Dorothy ranges up around 650. So they all think they're quite funny when they do that and original. So, just a little bit about where I came from. This is my family, my wonderful family. I grew up in Moundridge, Kansas. I have three older brothers who are much older than I am. I think I was definitely an accident when I came along. But this is my wonderful mom, Adelia Stucky. And this is our family farm, which is right near Moundridge, Kansas. And my brother Russ Stucky still owns and manages this farm. It's been in the family for about 5 generations. So it's a great place. Indeed, I was in a tornado that hit the farm when I was in the 4th grade. I was in one of these chicken houses over here with my mom because we were keeping our baby chicks moving around. So because they actually smother each other when they get too close to each other, they can't get oxygen. And so when we were in that chicken house, that little brooder house, the tornado actually hit our farm. It actually picked up the chicken house by about a foot and fortunately put the chicken house back down. So we did survive that, but it was a little bit of a narrow survival. But anyway, my mom always taught me to do a little bit risky things. So that was good. So this is my mom, Adelia Stucky, right here. She was the medical technologist for Moundridge Hospital, and she has really helped run that hospital. She was amazing, and she would let me come in and help her in her lab. So I basically grew up in that lab, and I loved being in the lab. She would let me, for example, wash out tubes of blood that she used to do blood chemistries on patients. And I remember being about, I don't know, 8 years old, and I just had this big dish pan of coagulated blood, and my hands were in there washing out these tubes. And how things have changed. I do not think that OSHA would approve of that these days. But anyway, I got 10 cents an hour to do that. So enjoyed growing up in that lab very much. And then grew up for a number of years in Moundridge and eventually went to this wonderful place called Bethel College. And I think a lot of you are familiar with this wonderful administration building. I had a wonderful experience at Bethel College. I guess I was there between 1983 and 1987. I graduated in 1987, and I wanted to bring up this fellow right here because some of you might recognize who that is. I believe that is the wonderful Dwight Crable, right? So Dwight Crable was absolutely instrumental in my career choice. I decided to get a major in biology and then a minor, almost a major, but a minor in psychology. And so Dwight really helped me get interested in the brain and in following a neuroscience path. Dwight, thank you so much for all of your support and encouragement all these years. It's been amazing. The other person I must give tribute to is the fabulous Dr. A. Wayne Weens. Many of you have had the great opportunity to learn with Dr. Weens. He really got me interested in molecular cellular biology. I took an advanced course when I was a freshman, and it changed my major because I was going to be a flute and English major. And at that point, I just got so enamored with molecular and cellular biology that I thought, this is the career for me, potentially. So anyway, while I was there, Wayne and Dwight allowed me to work on a senior research project using rats. And I should also say we had a wonderful outside mentor, Dr. Dale Horst, who helped with this project. But it was the first time I was able to actually do research. And I had an amazing partner in crime. This is Dr. Christie Neufeld. She is now professor of molecular biosciences at the University of Kansas, where she does groundbreaking work on cancer research. So she's finding new therapies for cancer, new ways to stop cancer. She's fantastic. So at that time, Christy and I were both in the same class, and we did this senior research project together where we took brains out of rats. So this is the rat brain, and you can see it's much less convoluted than the human brain, which I showed you previously. And we did a kind of fancy technique at that time where we did receptor binding assays using a instrument, a manifold kind of like this. And what we were doing was figuring out how many receptors for neurotransmitters like dopamine and norepinephrine were in the rat brain as it developed from neonatal ages on. So Christy and I really had a great time together working on that project, and I think the world of her. So then eventually after I graduated from Bethel, I eventually decided I just love the brain, love neuroscience. So I'm going to go on and go to get a PhD in neuroscience. And so I ended up at the University of Minnesota in beautiful Minneapolis. And as part of that program, the very first thing that they do in that program is immerse you in a neuroscience boot camp. And boot camp it is because you're up at Lake Itasca, Minnesota, which is actually the headwaters of the Mississippi. It's amazing. It's a beautiful lake. Anyhow, we were there for 6 weeks. We worked in the labs or took lecture up there from 7 a.m. till 11 p.m. at night every day. And so we were absolutely immersed in that. And this is my group of colleagues, my classmates who were there at Lake Itasca at the same time. And for example, Tim Gomez right here is one of my still wonderful colleague professors who's at University of Wisconsin-Madison. And then actually some of you might recognize this guy back here. This is actually Galen Kaufman, Dr. Galen Kaufman, who got a PhD in neuroscience. He's also a veterinarian, and he is from Newton, Kansas. So he's a good friend of mine. So what I learned from that experience of going through boot camp and going through a PhD in neuroscience is the competition is tough. Lots of people are smart, and there are frankly a lot of people that are smarter than you are. But all of that competition actually makes you better and stronger and more resilient. So then after I got my PhD in neuroscience, and at this time, while I was in graduate school, I actually met this other scientist who was a molecular geneticist who we ended up getting married. His name's Jeff Waring. And so we decided we'd always wanted to do something different in our careers. And we had always had, independently, we'd each had a dream of sometime living in Europe somewhere. And so we decided that we would apply for postdoctoral fellowships in Germany. And so we did that, and we ended up getting postdoctoral research fellowships in the same area of Germany. So this is the map of Germany. And we went to a little town, a small town on the Main River called Würzburg, Germany. And it's a beautiful town with a wall around it, a gorgeous castle on a hill. It's in the middle of wine country. So that was just a phenomenal experience. So here we are. This is my husband of now 27 years, Jeff Waring, and I were at the airport after we sold virtually everything we owned. We sold almost everything we owned, and we then packed up everything and went off to Germany. And here we are with our two kitties, and we took them with us. So we learned to take chances. So that's one thing I would like to say to you, that if you have risks and chances that are opportunities, you should really consider taking them, because you might regret not taking them more than you regret taking them. And so going off to Germany to do this postdoctoral fellowship was one of the very best things we ever did in our life. So we were adventurous. So we trusted our gut, and we took our friend, each other with us, and our cats with us. And then we ended up being there for 4 years. Why did we go to Germany? Well, I had, just speaking from my perspective, I had always wanted to learn how to record from live neurons. So I had done neuroscience as a PhD student, but I'd always worked with tissue that was fixed or frozen or essentially dead tissue. But neurons are so exciting because they fire action potentials, they fire impulses, and they do live excitable things. And I wanted to learn to record that excitability. So I wanted to learn physiology, essentially neurophysiology. So I love the idea of recording from live excitable cells because I'm kind of an excitable person. So I'd heard about this really unique technique called a skin nerve preparation. And I'll tell you more about that in a few minutes, but it was a fancy technique that no one in the United States was doing. And so there was this one person named Martin Kolzenberg who did this in Würzburg, Germany, and did it from mice, took out the skin and the nerve from mice and did recordings. And I thought, I'm going to see if I could get a postdoc with that guy, because if I did, I could bring that technique back to the United States and have like a niche of my own in my own lab. So that was why we went. Jeff had his own story of doing genetics in a different lab. But like I said, we always wanted to live in Europe, and both of us really kind of like adventure. In fact, we've always wanted to go back to Europe and live in a different place, maybe France or Italy. We haven't gotten that opportunity yet, but maybe a sabbatical will come along. So here I am learning electrophysiology in Würzburg, Germany. And here's my husband, Jeff, who happened to be stopping by the lab at this time. So this was we call these rigs, these electrophysiology rigs. And so a rig is just like all the components that you use to do your recording. It's the microscope, it's the chamber, it's where you put your tissue, it's the what we call the amplifier, the oscilloscope and the amplifier, all of that stuff together is your rig, right? And so again, why Germany? Well, Germans know physiology. They definitely know how to do physiology. They've been doing it for many decades, really. Also, Germans are quite disciplined, and it's very interesting to work in a German lab because although it's very similar to working in a US lab, I think the Germans tend to be more focused and more efficient with their time. They also have great senses of humor. So had a wonderful time there. So a little bit about that preparation. So what we do is we take a mouse. You can do this with a rat too, but you take a mouse and you essentially anesthetize the mouse. And then you can take out the paw skin, so the hind paw skin, either the top of the skin or the bottom of the skin, along with the nerve that skin innervates. So it's similar nerves to you. So you can take out, for example, the nerve that innervates the bottom of your foot, your plantar foot is called the tibial nerve. So you can take out the tibial nerve along with the skin, and it's fresh skin, you can put it into a bath, tissue bath, and keep it happy. And then you can take this nerve and you can tease it into very, very tiny little filaments until you can record from a single nerve axon. And then you can figure out where that axon projects in the skin. And then you can stimulate the skin with touch or cold or heat or chemicals or painful things, and you can record electrical activity or action potentials firing in that single nerve fiber. So that was really exciting. So I learned that there. Then eventually we did another postdoc in Berlin, but eventually applied for jobs back in the United States. And I was very fortunate to get offered a faculty job, an assistant professor job at the Medical College of Wisconsin, which is in Milwaukee, Wisconsin, beautiful city. And here I am in 1999. I have just landed in the lab. Here is me with one of my skin nerve recording chambers right here that I'm trying to figure out how to set up. I look back at this picture and I think, my goodness, I was young and wow, I was thin. Whoa. So yeah, lots of things have happened in the last 20 years. But one thing, so some things I've learned. I arrived to an empty lab. One thing I learned right away is you're rolling around in that lab, it's completely empty, and you like never thought about, how do you set up a lab? How do you even know what to order? I mean, well, who are you going to hire? No one taught you how to hire anybody. So you have to have, and also your data that you were collecting as a postdoctoral fellow, you were just churning it out. You were at the maximum productivity as a senior postdoctoral fellow. Well, guess what? When you start your faculty job, all of a sudden, like, there's no data. It comes to a screeching halt. So you have to have a lot of patience. I also had to learn to write a grant. So I didn't know how to write a grant. So I had to learn how to write a grant, and I'm still learning how to write better grants. I had to learn how to hire people, how to manage people. Sometimes I have had to let people go. I tend to call it, let me help you on a happier path. Let me help you to a happier career when I have to let somebody go. And it usually works out okay that way. Oh, sorry. I have had to learn to make lots of mistakes. There is no question that in any career you're going to make mistakes, lots of them. And one of my friends told me a very wise thing, that it's not about making mistakes, whether you will or you won't, because you will. But he said, Cheryl, it's how you recover from your mistakes that's the important thing. And that is absolutely true. So fast forward to 2021. And by the way, can we just skip 2020? Let's be honest. COVID has ripped us all a new one, and in so many different ways. And so it's been hard for everyone. It's been at every level. And I just empathize with everyone with COVID. So anyway, we're coming out of COVID now, I think. But what I had to do, or what do I do now? I've been in my lab for 21 years. My, how time flies, but what do I do? Do I do experiments? My lab would laugh if I said I did experiments. I can't remember when I last did an experiment. The best thing I do is in the lab is actually teach people to do that skin nerve recording that I showed you. I still train people to do that, but experiments went out of the realm of what I do right now. But I write a lot of grants, a lot of the time, and rewrite them. I edit a lot of papers for my students, and I edit a lot of grants. I expect my graduate students and postdocs all to write their first draft of every paper. I do not do that for them, but then we edit and edit and edit and edit. I teach medical students, first year, second year, fourth year medical students, all about the nervous system and the brain. And then I also teach graduate students pretty regularly. So I teach a lot of lectures. I had already 4 or 5 lectures this week. to both medical and graduate students. And then I do now a lot of other stuff. So I'm the director of what we call the Neuroscience Doctoral Program. So that's the neuroscience graduate program, PhD program at the Medical College. So it's the program like what I went to at Minnesota. So now I'm the director of this program. I've been doing that for about 10 years. I really enjoy it. That's actually a lot of fun. I direct the pain division of the Neuroscience Research Center. So I kind of help people figure out how to do pain experiments, how to collaborate with someone if they're interested in seeing if their disease model has pain or if their patients have pain. I help with that. I review a lot of grants. I chair a lot of grant review committees for NIH. And then I, for example, at the medical college, I now chair the neuroscience strategic plan for how neuroscience is going to grow at the medical college. So I'm quite excited about that because I guess that we're going to infuse many millions of dollars into neuroscience research at the Medical College of Wisconsin. So that's really exciting to help the college figure out how to spend that money. And then I manage people. Sometimes I manage people well, sometimes maybe not as well. I've made a lot of mistakes, but you learn as you go. So that's a big part of my job. This is my current lab, who I am extremely proud of and grateful for. As you can see, we channeled the Brady Bunch because we couldn't be all together during COVID to get a photo. So we all thought it would be fun if we took pictures of ourselves independently with our masks on. So here we have, I'll show you in just a minute who these are, because then not only did we take the mask picture, but we took the picture without the mask. So you can actually see everyone's smiles. So this is Kate Sadler, my most senior postdoctoral fellow, who is ready almost to become an assistant professor herself. Tony Brand is another postdoctoral fellow. Vanessa Ellers is another postdoctoral fellow. My graduate students are Alex Mikesell and Tyler Waltz. And then my lab manager is Tony Menzel. Amazing guy. And then my lab glue is the technician Ali Brzezinski. So we're a pretty diverse group. Really get along well. And that's one of the things I've learned is have a team that gets along really well, because happy people are productive people. Okay, so what do we do as a lab? So we work on a lot of different things, but the major areas are on mechanisms that cause chronic pain and mechanisms that allow us to sense touch. Like, for example, how you can pick up a glass of water without dropping it on your computer. I work on the molecules, the channels, the proteins that allow you to sense the glass to pick it up. So that's really, really exciting for me, as is the whole area of pain research. So I'm going to tell you about just a couple of projects to give you a flavor of what we do and how we do it. So this is the Stucky Lab. So our projects, just a couple projects I'll tell you about, are we have a pretty big NIH grant to work on sickle cell disease pain. And so I'll tell you about that disease. We have another really big grant, which is it's actually called a Javits Award. It is a 7-year grant from NIH, and it's named after Jacob Javits, who was a senator in New York who died of ALS, or Lou Gehrig's disease. But anyway, we were awarded with that because it's a rare disease. I won't speak too much about that today, but I'm happy to answer questions later about it. We also work on touch sensation, again, how we sense things you pick up and text and all that. So that's through skin cells called keratinocytes and how they're involved in touch sensation. So we have a NIH grant on that. And then we're always looking for new drug targets for inflammatory pain and nerve injury pain, and even migraine. We're starting to work on migraine pain now. And also we're starting to work on cancer pain. So cancers are treated with chemicals called chemotherapeutics. And those chemotherapeutics have a side effect where they cause the sensory neurons to get damaged and die back. And that causes the patients excruciating pain. Sometimes that causes patients to need to stop the cancer treatment of the chemotherapy. So we work on cancer chemotherapy-induced pain also. So one of the things we work on, like I said, is sickle cell disease. Sickle cell disease is the most common inherited blood disorder in the US. It affects about 1 in every 400 African Americans. But one thing people don't often know is that it also affects people who have descent from India, from the Mediterranean, and from the Middle East. So we can, and also Hispanic populations. So it affects more than just African Americans. It affects around 100,000 Americans in the US. And that ends up being about 3 million people worldwide. And there are some regions of, for example, sub-Saharan Africa, where upwards of 30% of the population in those regions has sickle cell disease. So it's a really terrible disease. What causes it is there is in the genes in hemoglobin. Hemoglobin is the molecule that carries oxygen around to your blood system and your brain. In the hemoglobin molecule, there is one gene has a point mutation in one amino acid that makes up that gene, just a single point mutation that is mutated, and that causes the red blood cells, which are normally like little inner tubes, right? Real flexible inner tubes that move in and out of your vessels and your capillaries really easily. Well, this point mutation causes the red blood cells, when there's low oxygen, the people are exercising, or they get stressed, or they can't breathe, that causes the hemoglobin molecule to make a really long polymer, a long stringy polymer, okay? And it's sticky, and it causes this red blood cell to now become sickle shaped, like a boat, like that. And what happens is now those red cells cannot move easily through the vessels and the capillaries, and they get stuck and they pile up on each other, and that's called a vaso-occlusion. And that then causes the oxygenated blood to not be able to get to the distal tissue, and that causes acidosis and ischemia, and that is the problem. And then there's also, it's an anemia disease, because these red cells are so fragile that they fall apart, they lyse and fall apart 10 times as easily as normal healthy red cells. So when they lyse, they dump all their contents into the blood, and that causes the anemia. So they no longer have enough iron and enough hemoglobin in the red cells. Okay, so why are we interested in this? Well, it's really important because it actually has been known for many, many years by doctors and physicians who treat patients with sickle cell disease that they have very severe pain. But people who were researching pain didn't know this until maybe 10 years ago. Many people still don't realize this, but people with sickle cell disease have very severe pain, especially when they're in one of these, they sort of call them crises, but when they're in a occlusion, when the red cells are occluding the vessels, that causes severe pain, causing them to go to the emergency room and often become hospitalized. And they often also get chronic pain even when they're not in a sickling event. And so babies start getting these episodes of sickling by the time they're about a year of age. And what happens is, in babies in particular, is they get this swelling of the hands and the feet. It's severe swelling and edema in the hands and feet. It's called dactylitis. But anyway, the swelling causes them to have excruciating pain in the hands and the feet. And then as they progress and they grow up, they continue to have these episodes and they continue to get painful episodes and develop chronic pain. This right here is an artist named Hertz Nazaire. And Hertz has severe sickle cell disease. And he decided to express his pain with sickle cell disease through his art. And he's done a number of really interesting paintings that some of them are shown here. But this one I always think is really very interesting because it is his, he calls it 10 redefined. And the reason for that is that if you're ever asked about your level of pain, you will give your answer on a scale of 0 to 10, with 0 being no pain at all, to 10 being the most severe, horrible pain you have ever felt in your life. So that's the scale. This is Hertz's depiction of what his 10 feels like. It is horrific. It is awful. The patients describe it as feeling like knives are just sticking your body all over, and knives, and it's horrible. It's awful pain, and and they can't do anything but go to the emergency room and try to get help. And one of the problems that patients that have sickle cell disease have is because of the demographic that they come from. So many are African American. When they go to the emergency room, they're looking for pain relief. They're asking for an opioid or a narcotic because they know that when they're in an episode, that's the only thing right now that's known to relieve their pain that they can get. But often, because of the demographic they're from, they're considered drug seekers and they aren't getting their pain relieved. So it's a very a sad area of discrimination, and we're working hard to try to alleviate that level of discrimination. So my lab also then works on what actually causes the sickle cell disease pain. So how can we get non-opioid, non-addictive therapies to alleviate sickle cell disease pain? So my team members are postdoc Kate Sadler, postdoc Vanessa Ellers, and then this woman is Amanda Brando, who is an absolutely amazing physician who treats children and adults with sickle cell disease. So she's a clinician at the Medical College, and she is really instrumental because she guides all of our work that we do in mice and rats. These are called preclinical studies, with how the patients feel. She tells us like whether we're doing the right kinds of assays for pain. And she helps us also get samples of blood and serum from the patients that we can test out in our pain models. So she is amazing. Now, what we use then in our lab largely to study sickle cell disease is mice. And how can we do that? Well, there are mice that have been genetically altered so that they've had their mouse hemoglobin knocked out or deleted from their genome. And in place of that, we've put in human hemoglobin. And so we have mice that have normal human hemoglobin, just like you and I have. And then we have mice that actually have sickle cell hemoglobin that have that point mutation that I described in patients. And so the mice amazingly have a very similar phenotype to what occurs in the patients. So these are some of their sickled red blood cells. They have anemia, and they, when their red cells sickle, they clog the vasculature. Here's a vascular occlusion example, and that causes ischemia because the blood, the normal blood carrying oxygen, cannot get to the distal tissue. And so these mice look just a lot like patients. And so how does one study pain in a mouse? What a good question. We're still trying to figure that out. But one of the things that we do is we put the mice on a tabletop, and we put a glass top over the mouse, and we can activate the bottom of their paw with different kinds of touch stimuli, these filaments that are calibrated to elicit a certain force when they bend. So we can get what we call thresholds for sensitivity in the mice. We can also do thermal stimuli of the bottom of the foot. We can apply things like cold through dry ice to a glass table that the animal is sitting on. So it doesn't freeze the animal's paw, it just cools the glass underneath the animal's paw. But we do these kinds of evoked stimuli to measure withdrawal latencies or withdrawal thresholds and get an idea of their sensitivity to pain. And they do express, they're extremely sensitive to touch. They're extremely sensitive to cold. Excuse me. Now, one thing though is that patients that have sickle cell disease, and any kind of patient with chronic pain, often has not just evoked pain, but spontaneous ongoing pain. And how do we measure that? Well, we do this through a system called a home cage scan, where we can put the mouse in its home cage, and then with a fancy video recording apparatus, we can measure all kinds of things that the mouse normally does. We can measure, for example, how does it rear up? How does it hunch over? How does it move and how fast? Does it try to climb up on the roof of the cage or not? Will it hang from the top of the cage? Does it build nests? And all of that can be used and automated through this video system to figure out if the animal's moving less, which might indicate pain, or if it's hunching over, which tends to indicate pain. And even one of the things it can do is measure the eyes being pulled back in a sort of grimace, because grimace is one of the characteristics of an animal in pain, when it flattens its ears and its eyes and it pulls them back. So we can see if they have ongoing pain. And then how else do we study sickle cell pain? Well, we study it by taking out the sensory neurons and the pain receptors. So excuse me, I'm going to tell you just a little bit about that. And I'm going to illustrate what we do in a mouse by showing you what it looks like in a human, in you. Here's your spinal cord, right? And here are your ganglia. These are dorsal root ganglia. That is a ball of neural tissue that sits just outside of your spinal cord, on either side of the spinal cord, at every level, cervical, thoracic, lumbar, and sacral. And so if you blow up, expand what that actually looks like, here's your spinal cord, and here is this ganglia of tissue. And the reason that's important is because the ganglia has about 20,000 cell bodies of sensory neurons that go out and innervate your hands and your muscles and your feet, everything that senses your external world. And the cell bodies are largely made of pain receptors. So what can we do with that? Then we can take those neurons out, and we can basically dissociate them up and then plate them on a cover slip and put them in culture. And so here is what one of the dorsal root ganglia neurons looks like. And this is just what one of your sensory neuron dorsal root ganglia would look like under a microscope, where it's magnified many times. This is a beautiful cell body of the pain receptor, of a pain receptor. And if you apply antibodies, you can antibodies to stain different proteins and label different proteins on the cell body, it looks like this. So here's the cell body, and here are axons coming off of it. So what we do with that is then we take those neurons, and we can do something called a fancy technique called calcium imaging. And all that is we take the live neurons that are growing in culture, and we can put a calcium sensor into the neuron. And calcium is one of the big neurotransmitter molecules that is elevated when pain receptors are activated. And so then we can record under the microscope and we can apply itchy things like histamine or hot pepper things like capsaicin, and that will elicit a firing, an active activity in that neuron. Another type of experiment that we do is called patch clamp recordings, where we can take that same cell body neuron that I just showed you, and you can patch clamp onto that neuron membrane with an electrode underneath a microscope, and then we can poke the neuron's cell body with a mechanical probe with a very tiny amount of force driven by a piezoelectric device. And this is a kind of response we can measure in that neuron. It's a current when a mechanotransducer channel is opened. So we can record from these neurons, we can look at the channels that underlie pain responses. And so here is another way we can look. One last technique I'll tell you about is we can take the animal's paw, and that's that skin nerve recording. We can dissect out the bottom of the paw and the tibial nerve that innervates that paw. And here's this example that I showed you earlier, where we keep that keep that tissue happy in the bath. We record from a single tiny axon in that nerve, and here's what those nerve firings look like. And so we can record all kinds of different activity. So what are we finding? Just one thing we found I just want to tell you about is very cool. If any of you like hot chili peppers, like I like hot chili peppers, these are habaneros, right? And Thai peppers and all. What makes the peppers hot? It is a very lipid-like molecule called capsaicin. Capsaicin is the molecule in all kinds of hot peppers that makes it feel burning hot. We found that the receptor, so the protein, the ion channel port in the pain receptor is what is underlying sickle cell pain. It is the capsaicin receptor. And here is just a schematic of what that receptor looks like. And that receptor is activated not only by capsaicin, but also by protons and by heat. And now we're finding it's activated by lipids that are circulating in patients with sickle cell disease, their serum. And so we're studying now the endogenous things that are in patients' serum that activate this receptor. So we're hoping to figure out a way in which we can target this receptor in clinical trials for sickle cell disease. And one other project I just want to briefly tell you about is that we're also working on how skin cells convey pain and temperature. This is the Gunther von Hagen's display of the man holding his own skin. And I really like this because it shows you just how huge your skin is. You know, it's the largest organ in the body. It actually covers, if you stretched out your skin, it would cover over 20 to 22 square feet, and it weighs like 16 pounds. It's amazing. It's huge. It covers almost all your body. And for many years, until recently, it was thought that the skin's main function was to keep bugs and bacteria and viruses out and keep moisture in your body. But we are now figuring out through the lab's work that these skin cells, the keratinocytes on the outside of your body, they actually respond to temperature and painful stimuli. So it's not just the sensory neurons that are embedded in the skin, but it's actually the skin cells themselves that respond. And this is my team member, Alex Meixel, who's working on this project. And we've now taken skin cells, these keratinocytes, from mice paws, from rat paws, from a 13-line ground squirrel, which is a obligate hibernator, which is really an interesting animal I can tell you about sometime, and then human skin cells, and found that they respond to temperature, and they respond to touch. And our lab has just recently identified, and we are just about to publish this, that the way in which keratinocytes respond is by activating this channel, this protein port in the keratinocyte membrane called piezo-1. Piezo stands for, it means Greek, or it means pressure in Greek. So it's piezo-1. It's a gigantic channel it's just the hugest channel that's known in the body. And so we just discovered that. So that's really exciting. And so what's next? As I close, our mouse is asking, well, what's next? The one issue we've had in the pain field is that many targets have not actually, that have been found in rats and mice, have not actually translated to the clinic. So to actually being able to be targeted in patients. And why is that? There are a number of reasons that might be the answer, but one thing we're asking is it really a species difference, that mice and rats are really just different than humans? And so question is, how can we test this? So what we're now doing is we are able to surgically extract human dorsal root ganglia from organ donors and also from people having surgery for tumors that impinge on the spine. So for example, if someone's having a spine tumor removed, often the surgeon has to take out the dorsal root ganglia as well, and that is usually discarded. And so we're asking our surgeons if we can get that fresh ganglia tissue. And so here's the spinal cord, the vertebrae, the dorsal root ganglia, and the human, and we're excising that, our surgeons are. Here's what that tissue looks like. Here is the ganglia right here and the nerves. We cut those nerves, and here's the human ganglia, what it looks like actually in a dish. And so here is when we take a cross section of tissue through that ganglia that's fixed, and you stain it with different antibodies, you can see the cell bodies of these neurons that have their cell bodies and then their axons. These are all the axons that are going out to the peripheral nerves. So you can just see all these beautiful, very different sensory neurons. Many of these are pain receptors within this human dorsal root ganglia. So what we're doing now is partnering with spine surgeons at the Medical College to obtain human dorsal root ganglia. And I just presented to them 2 days ago, and they're on board to do this. So they're going to be taking tumors out of patients near the spine. And again, those DRGs often have to be removed and thrown away, but they can give us those DRGs if the patients consent, of course, to having their ganglia used for an experiment. And the patient's pain can be assessed and evaluated before they go into surgery. So we can get pain phenotyping along with the responses of the neurons. And in addition, we're trying to figure out how to remove dorsal root ganglia from cadavers after the teams have come in to harvest the organs for organ transplants. And so what we can do is, again, do those same recordings that we did in mouse and rat neurons, but do them on human neurons. And we're one of the labs in the world, in a few labs in the world that does this mechanical current recording. And what I'm really excited about is nobody's done this in human neurons. And so we're going to hopefully be the first to be able to assess how we sense touch in a human sensory neuron. And so we're just looking at a lot of different types of chronic pain. All of these different types, we're getting into migraine pain, chemotherapy pain, and so forth. Let me just ask, Dwight, would you like me to wrap up real quick here? I'm almost done.
Just go ahead, it'll be fine.
Okay, thanks. Great, thanks. So I just wanted to share a couple of thoughts after 21 years, what helps to find success, find an area you're passionate about, enjoy it, and connect with others who share your passion. And do not be afraid to try new directions. Again, take some risks. You'll be happy you did. Think about unmet need in research. Where can you find the most unmet need that you're passionate about, and figure out how you can capitalize on this, and get into these research questions. I would say that you want to learn to collaborate well. You want to explore novel techniques that will actually answer your question in a state-of-the-art way. If you go into this career, you need to learn to write really good grants, but you can do that. You can find expert consultants. You can hire a grant writer. You need to publish regularly. Smaller papers are fine when you first start out. Don't be a perfectionist, because perfection can lead to procrastination. And that's probably true for most careers, but I have learned this over and over. Do not be a perfectionist. What I've learned, again, after 21 years, have patience. Really, this is really important. You do not have to have it all at the same time. For example, we had 2 kids during this 21 years, and it was at times, stressful trying to figure out how to manage babies and manage toddlers, as we had 2 science careers going. What I learned later is you can have different things at different times, and to have patience. One of the best things about this career is you get to work with smart, young, really interesting people that are really creative and have great ideas. Science is never boring. It's always changing. It's always interesting. You always learn new things every single day. Any kind of science that you're in, you learn new things every single day. Have compassion for others. Have compassion for your students. And when you get where you're going, like the Tim McGraw song says, reach behind and help the other person up. So try to pay it forward to your students and your trainees and others who are around you, because you want to help train that next generation. And that is the most wonderful thing, is to see people who come after you be successful. Your positive influence on those around you will have a very big impact, and you will not even know how or why, but it will. You want to help build others' confidence. And that's something I do every day. I am working on these trainees' confidence to build them up. It is way more important to seek the truth and be sure about it than to get a Nature paper. And I learned that over and over and over again. We have to be rigorous in seeking the truth. And that's way more important than a flash in the pan Nature paper. I learn more from others than they learn from me. And again, it's not about making mistakes. It's how you recover from them, because you will make mistakes and you will have to recover from them. And that's what it's all about. And so with that, I guess I'm still following that yellow brick road, looking for, I guess in my case, I would really love to do something that would ultimately help a patient alleviate their pain. That is still my goal. I haven't achieved it yet, but I hope that in my lifetime I can do something that will really help patients. And with that, I want to thank all of my lab members, postdocs, my 4 postdocs here, graduate students right here, in my lab manager and technicians. And then I really want to give a shout out to my physician scientists who I partner with all the time. Amanda Brando, who works on sickle cell disease. Gwen Hoban is a plastic surgeon who she actually helps us get patient tissue. For example, if someone's having a leg amputated for chronic pain, she helps us garner that tissue. And then our head of neurosurgery, Shaker Kirkpad, is helping us get connected with the spine surgeons and get excited about getting human tissue. And then my funding sources are from the National Institutes of Health. I'm extremely grateful for them. And then I also get funding from Advancing a Healthier Wisconsin. And finally, I want to most of all thank all of the rats and the mice that have given their lives to this research. And I would like to thank the patients in particular who have given their lives to this, or not their lives, but given tissue to this research. And hopefully we can help them in the future. Thank you very much for your attention.